Click the bell icon and subscribe to our channel so that you will not miss any classes. After completing meiosis 1, I told the cell will spend some time in interkinesis where it will rest. Because we are calling it as interkinesis, there is no DNA replication. Initially, when we were studying the cell cycle, we studied that interphase means there is a replication of DNA, but here there is no DNA replication. Hence, we are calling the term interkinesis. After completing the meiosis 2, there is formation of two daughter cells as we studied. Two daughter cells are formed. We have started with one cell. And here there is formation of two daughter cells after completion of meiosis, and each of them are having n number of chromosomes, and the chromosomes are just not lose their integrity. Now when I will take one cell and I will explain how it completes the meiosis 2. Now the cell will enter into prophase 2. In prophase 2, what happens? The nuclear membrane is started disintegrating. There is a separation of centrioles with the formation of spindle asters, and there is a thickening of the chromosomes. And here for thickening of the chromosomes or for the condensation of chromosome, much time is not required, because I told in the telophase 1 that there is no complete dissociation of the chromosomes, uncoiling of the chromosomes not taking place here, because and the chromosomes have their integrity, at least integrity will be present. So for thickening it doesn't require much time. So there is a thickening of the chromosomes has taken place, that is the condensation of chromosome taking place, and there is a disappearance of nuclear membrane. Nucleolus will disappear, and the associated cell organelles with the nuclear membrane will also start disappearing. By the end of prophase 2, all of the process will complete, and later the cell will enter into the next phase of division, that is metaphase 2. In metaphase 2, what happens? The chromosomes will arrange in a equatorial plate, that is there is a formation of cell plate. Cell plate is formed, and there is an attachment of spindle fibers to the chromosomes at kinetochore from the opposite poles. That's why, this is the reason why we will say same as mitosis. Meiosis 2 is same as mitosis. Only the difference exists in meiosis 1. Meiosis 2 is exactly similar to that of the mitosis. And now after attachment of the spindle to the kinetochores on the opposite poles, then it will enter into anaphase 2. Anaphase 2, where the chromosomes are split at the centromere and they start moving to the opposite poles, and we may observe different shapes of chromosomes as we studied in mitosis, depending on the type of chromosome, metacentric, submetacentric, telocentric, acrocentric. Depending on that, we can see the different phases of chromosome, different shapes of chromosome as L shape, I shape, V shape, J shape, like that. And after completing anaphase is the telophase. By the end of anaphase, the chromosomes have reached their opposite poles, and in telophase 2, the chromosomes are reached to the opposite poles, and there is an appearance of nuclear membrane and nucleolus, and there is a complete dissociation of the chromatin. Chromatin is got dissociated, and the cell will appear like it is having two nuclei in a cell. To compensate that, the cell will divide its cytoplasm, which we call it as cytokinesis, and after cytokinesis there is formation of two daughter cells like this. Two daughter cells are formed, and these daughter cells are having how many number of chromosomes? N number of chromosomes. And how many cells are formed here? Two cells, right? And we have taken only one cell here, and there is a remaining one more cell. The same process will happen. As a result, there is formation of two more cells. Two more cells will be formed here, and they also having n number of chromosomes. So now you got how four cells are formed in the meiosis. In first meiosis 1, there is formation of two daughter cells, and these two daughter cells will enter into meiosis 2. And don't think one after the other they divide. No, simultaneously the process is happening. So I just taken one cell to explain. So here the two cells will enter into the meiosis 2, which is similar to that of mitosis, and finally they have got four number of cells which are haploid. Meiosis is started with diploid cell, and here it is ending with four daughter cells which are having n number of chromosomes. This is how the number of chromosomes are reduced in meiosis. So this is about the stages of meiosis, and one more thing to be discussed here. There are two types of the cytoplasmic division or cytokinesis. They are successive cytokinesis, and the other simultaneous cytokinesis. Successive cytokinesis means after meiosis 1, the cytokinesis will take place, and it is followed by meiosis 2, and again cytokinesis will take place. This is called successive. That is, after completion of meiosis 1, the cell will divide once, and after completion of meiosis 2, the cell will again divide. That is its cytoplasm. And simultaneous type is after meiosis 1 and meiosis 2. After meiosis 1 and meiosis 2, the cytoplasmic division, that is cytokinesis, takes place at a time. That is, after meiosis 1, the cytoplasmic division will not take place. Totally together, after meiosis 1 and 2, the cytoplasmic division will take place. So that is called a simultaneous. And one more thing, in meiosis 2, the number of chromosomes are carrying the same, because after completion of meiosis 1, the cell have got n number of chromosomes, and the same number of chromosomes are carried to the daughter cells also. Hence, the meiosis 2 is called homotypic division. Whereas meiosis 1 is called reductional cell division, because the number of chromosomes are reducing to half. Because meiosis 2 is a part of meiosis, we are not calling it as an equational division, we are calling it as a homotypic division. But mitosis, we will call it as a equational cell division. So that's all about the meiosis and how the 4 daughter cells are formed in the meiosis. We shall discuss the significance of meiosis in the next class.